Cool. Hello, welcome. We're at Microbiomes for All, co-sponsored by REMNet and ASM Education. My name is Rachel Horak. I'm coming to you from my home office outside of Boston, Massachusetts. I do programs related to undergraduate education for educators, including the ASM Conference for Undergraduate Educators, which will be held in Phoenix this year in November. Our speakers for our series will be Theodore Muth and Davida Smythe. And this is an activity as part of their NSF RCN-UBE grant, and they'll tell you all about it. So during the webinar series, we have a webinar every 2 weeks, and the recordings will be posted within a business day. There'll be occasionally some work to do between the webinars. You'll hear more about that later. And you'll access all of the content for this webinar series through our LMS. And that's how you got here today. So use the same link for all of the activities for this series. Now, all the handouts and slide decks will be in the Google Drive folder. And the link to that was in the LMS. And when I get done talking, I'll post the link also to get to the Google Drive folder. Okay. So if you have questions about the logistics for this series, you email me at education@asmusa.org. If you have questions about the content, you're going to email Davida and Theodore, and they'll share that information with you in a bit. Okay. So if you have questions, if you have technical difficulties, let me know throughout the session. I'll be here monitoring the room. Otherwise, Davida and Theodore, thank you so much for being here, and I'll let them take it from here.
Hello, everyone. I'm Theodore, and I'm joined here by Davida and Gina, who are all part of the leadership team on our REMNet RCN-UBE. And let me see if I can share my screen here. Alrighty, and all right. Can everyone see everything all right? Okay. Right. So this is the first session of a four-part webinar series focused on data analyses. And just for a little bit of background, like I said, REMNet is an RCN-UBE. It's research experiences with microbiomes. And sometimes we just refer to ourselves as Microbiomes for All, since that's kind of our mantra and our goal. And we've worked with probably at this point a few hundred faculty or more on integrating microbiome research, oftentimes as course-based research as a cure, but not always as a cure, making these resources available to faculty and then providing support as we go along. And when we get more towards the end, we'll also say that we're very much relying on the community, and we really encourage everyone to share their expertise, their insight, and their enthusiasm, and be a part of the REMNet team and kind of help us out, because we are really kind of relying on the expertise, drawing from a lot of experts in the community. So if you're here, you probably already have a good idea of why microbiomes are interesting. We started this work a few years ago at the City University of New York. I'm here at Brooklyn College. Davida was my CUNY colleague for a few years, but then she abandoned me. But we're still carrying this out at CUNY. And again, like I said, REMNet resources and tools and microbiome projects are being run at many, many other institutions across the country. We focused initially on urban microbiomes because those were largely understudied and still are largely understudied. So you can see here pictures of our students collecting samples from around New York City and then processing those in the labs at CUNY. I've just kind of made this slide. I know it's a little texty, but in terms of, if I could actually see my slide, it would be helpful. One sec, if I can move this thing around. Yeah, okay. So these are general types of microbiome research projects, and they're kind of ordered from kind of entry level to more complex, involved ones. The easiest ones to do are surveys, like we started out kind of seeing what's in the environment, what's in the built environment, what's on the sidewalk, what's on the subway. And this can be done by collecting, water samples, soil samples, swab samples. And, you can get a lot of distance out of a few samples. It's a fairly easy way to kind of enter into this. If you want to go up a notch, you can do some comparative studies where you're collecting samples from different environments or different locations, or you might set up things in the lab. So you take one set of samples and expose them to, 37 degrees, and another you expose to 25 degrees, some type of environmental variable, and then you compare the microbiome communities before and after those exposures to stress. And this, again, allows a little bit more for some hypothesis development. It's a little more involved, gets the students able to kind of think more scientifically. And then you can incorporate animal, plant, insect microbiomes. This is always kind of very exciting. And again, you can do this either kind of as a survey approach or as a comparative study. And then again, you can do microcosms or mesocosms and really work on controlling the different variables. Some excellent work has been done with Winogradsky columns, looking at microbiome communities there. And maybe near the end, I'll talk a little bit more about some of the work that we've been doing with synthetic bacterial communities and looking at those. So lots of possibilities. This is just kind of a little overview of some of the broadest categories. And just, if I didn't mention, if anyone has a question, I think if you raise your hand, hopefully you'll pop to the top of my little celebrity Hollywood Squares thing, and I'll see you. But jump in if you have any questions or want clarification as we're going through. So this is a table from a publication from a couple of years ago, listing lots of great examples of published work with microbiomes and students. So I encourage you to check these out. A lot of them will give you nice kind of tools on how to approach this, and ideas of like how you might be able to integrate microbiome research into your courses. And so I think now, Davida, can we do the little videos? Okay, we have a couple testimonials.
All right, I hope you can all see this. All right, here we go.
Hi, my name is Christine Gertain, and I'm the director of a 3-year authentic science research course at Toms River High School North and Toms River High School South in Toms River, New Jersey. Our ASR students do DNA barcoding projects looking for the presence of a bacterium called Wolbachia, which is found in arthropods. DNA was extracted from mosquitoes in New Jersey and in Israel to search for the presence of Wolbachia in partnership with the Bordenstein Lab at Vanderbilt University. Students looked to schedule meetings across time zones, worked with partners that had English as a second language, and they learned that Israeli students attend school Sunday to Thursday. It was a true global experience. I also had 2 students work on soil microbiome projects. One was aquatic soil samples. The other project investigated the effects of insecticides on microbes in soil samples while they were growing ryegrass. The students used DNA subway to analyze their metabarcoding results. 3 sisters did research working on different types of oral bacteria related to tooth cavities. They each investigated different molecules that would inhibit bacterial growth, measuring zones of inhibition. In January, I will be hosting a bioprospecting training course that will train and certify teachers to run a bioprospecting research lab in their high school, where students could learn how to discover novel antimicrobial candidates.
Scene is amazing. Okay. Do I get out of this one? First one, second one, full screen.
Hello, my name is Carlos C. Galler, and I'm an associate teaching professor at North Carolina State University in Raleigh, North Carolina. I teach undergraduates and graduate students molecular biology techniques and skills, including bioinformatics and wet lab skills. And in 2013, we developed a metagenomics class. And we started by sampling kitchen sinks, and then we went across the campus sampling odd environments like downspouts, drains, water fountains, to learn about the microbial communities that are around us. Our research question is, what interesting microbes are around us that we often overlook? What features do they have? What genetic potential do they hide that we can take full advantage? And I use several different pipelines with students, including Chime2, Nephele, and Keemei. So far, we found some exciting microbes and some connections between the environment and our built environment and microbes coming back and forth. Thank you. Take care.
Okay. And our last one comes from Alice.
I am Alice Taron, and I'm an assistant professor at St. Lawrence University. I teach undergraduate students in general biology, and I also conduct research with students. My interest is on soil microbiome analysis, particularly in their application to sustainable agriculture and bioremediation. I have conducted projects looking at the ability of wetlands to clean up groundwater contaminated with heavy metals and petroleum, and how a series of wetlands and aquifers are able to remove these contaminants before they are released to water. So I am interested in how microorganisms in the wetlands may be responsible for this cleanup of contaminated water.
Back to you.
Great. So hopefully everyone can see that there's lots of really exciting projects out there, and even am I in the? Sorry, I'm sharing the wrong screen. And even in high schools, it's again easy enough to carry out there. So, all right, can everyone still see my screen now? Okay, beautiful. So one of the other things to think about, or I guess when you're considering if a microbiome project makes sense, is they're really flexible in aligning to a number of curriculum objectives or learning goals, and excellent for integrating in quantitative reasoning and data analysis skills. In this figure, what you can see is that the core elements of a microbiome project are listed down here in the center in blue, from experimental design to sample collection and so forth. During this webinar, we're obviously focusing on the data processing and analyses, but I've just shown as examples that you can align each of these steps with various kind of either quantitative objectives or other. These are all curriculum alignments for microbiology related learning objectives, but you can do it for ecology or genetics, intro bio. So there's a lot of flexibility, and pretty much each step of these projects, you can get kind of a lot of bang for your buck and hit the objectives that you're looking for. So we're going to do a little bit of a participant involvement thing. Am I turning it over to you now, Davida?
Yeah, sure. So in essence, I've put the Jamboard link in the chat. If you all go there, And we have 4 prompts. Each Jamboard page will have a different prompt on there. And if you all take like the next 5 minutes or so, not even, to just fill out what you can on the Jamboard.
2 minutes.
2 minutes. Yeah. Here's the link in the chat just in case you can't find it. I see some of you already have started.
And Davida, you'll share the Jamboard outputs, or I need to do
I can do it right now. Give me one
Okay.
second. Okay. So if you haven't used Jamboard, folks, if you look to the left-hand side here, there's a little sticky note here. If you all see this, there's a little kind of a sticky note. I'm hovering my mouse over it. You'll all love this. If you click on that, it'll make a sticky note, and then you can add your your words. There you go. Which sticky note is the one that's in the middle of the okay. And you can even change the color and all of that. You can move them around. So if you all fill out a sticky note, and then as I said, each page has different prompts. Okay. And we may not get through all of these today, but we'll at least start with the first one. What are you interested in looking at?
These are great. There's lots of good stuff with C. elegans that you can find some great protocols already out there. Soil microbiomes, you can talk to us. We're experts with that. Microbiomes of fermented foods, fantastic. eDNA, again, yeah, so depending on where you're coming from, you may be referring to things as eDNA or environmental DNA. We usually just talk about microbiome DNA, but essentially it's the same thing, but lots of great eDNA projects out there.
Great.
Wow, okay, lots of, yeah, Winogradsky columns, those are excellent. Wow, lots of great ideas out here. Oh, fish guts.
Delicious.
Do we get to eat the non-gut part?
Yeah, exactly. I think you learn too much when you start looking at what you eat. So what we'll do is while you're working on your other parts, we'll curate this and we'll put you into groups. You can kind of see what the diversity of the samples.
One thing while we're looking at this that I'll say, just to keep in mind if I forget to mention it later on, is that like this campus urban microbiomes, we've done a lot of swab samples. It's a little more of a challenge when there's not a whole lot of bacteria in your sample. So oftentimes if you ask students what do they want to test, you might hear things like, oh, they want to test cell phones or keyboards or bathrooms. And you can do that, that's fine. It's just that there's not as much DNA there. So it kind of sometimes can get a little bit trickier. Obviously, if you're doing something like soil or water, there's usually tons of bacteria there. So just something to keep in mind. All right, this looks great. So I guess we will curate this and share it later. Okay. All right, so just as a bit of an overview, like I said, today we'll get started with getting your sequence files into Nephele. Nephele is one of the tools that we'll use. You heard Christine mention DNA Subway. You heard Carlos mention Keemei. There are a bunch of resources out there. We mostly work with Nephele because we think it's really easy and it's free. But other tools are out there as well, and they work generally in a very similar fashion. So today we'll get started. Next week, we'll get more into alpha and beta diversity and other analyses. And then we'll continue with that and get to some more exciting visualizations. I didn't put part 4 up here because I'm going to circle back to that at the end and ask a few questions of you so that we can tailor session 4 to what people are most interested in. Okay, so let's go. So I'm going to show you first the microbiome pipeline, the analysis pipeline. And these, again, have the major steps, starting with your complex bacterial community. I'm going to default to bacteria, but again, you could do fungal communities with an ITS gene. You could do eukaryotic communities with an 18S gene. So most of what we're going to show you, you don't necessarily have to do it with bacteria. There are other options out there as well. So again, then extracting DNA, sending that out for sequencing. We'll talk a little bit about some of those options. And then you're going to get back, maybe some of you already have FASTQ files with lots of sequence data. And so we're going to kind of pick up here where we take this data, and we're going to use QIIME2, which is built into Nephele, to get some really interesting information from those sequence files. And hopefully in sessions 2 and 3, show you how to develop some of these visualization tools with your students. Okay. Any questions with the general kind of pipeline before I move on? Okay. So I throw this little thing of Repo Man up here because it's one of my favorite movies. But if you've ever seen Repo Man, you know it focuses on cars. And I put this up here because I don't know very much at all about cars, but I can get from point A to point B driving in my car with no problem. And there are lots of complex steps going from your complex microbial community into these, until you can get to these visualizations. And you don't need to be an expert at each of the steps. You don't need to necessarily know all of the little details that are going on underneath the hood, so to speak. And you can still really get a lot out of this. So this is a reminder not to be intimidated or scared off if you're saying like, well, I don't necessarily get all the details. That's fine. You can still really get a lot out of these microbiome projects. Some of the things that you should consider, I mentioned a little bit of this before, where to sequence. So we have been doing a lot of Illumina sequencing at Wright Labs and Mr. DNA. A lot of other companies, GeneWiz, which I think might now be Azenta. You can also do microbiome sequencing there. Usually it seems like once or twice a month, I get an email from another company that's offering microbiome analyses or sequencing, and sometimes with or without analysis. We've had a lot of success with Wright Labs and Mr. DNA because they don't mind working with small groups like us and taking kind of crazy questions and so forth. And I think if you reach out to Wright Labs and tell them that you're associated with REMNet, there may be a bit of a discount on the cost, which is, again, generally, if you're doing standard 16S sequencing on a single sample, it's going to be, depending on how deep you sequence, $50 to $70 per sample. Like I said, we do mostly Illumina, but in session 4, we'll talk about Oxford Nanopore, and maybe some of you others might want to add your experiences if you've been using Ion Torrent or PacBio. But we'll talk in this webinar about Illumina and Oxford Nanopore. I mentioned we're focusing on 16S, but you could do 18S or ITS. You can do whole metagenome shotgun. There is a pipeline for microbial metagenomes in Nephele. We're not going to talk about it during this webinar, but that's also an option if you're not doing an amplicon-based project. And again, you could also target other genes. If you're really interested in cellulose degradation or the nitrogen cycle, you can adapt the basic steps to look at the gene family that you're interested in. And like I kind of alluded to a minute ago, depending on questions that you're trying to ask, do you need 1,000, 10,000, 100,000 reads per sample to answer the questions? Those are some of the things that kind of go into planning and can affect the cost and how many samples you can afford to sequence. And the other thing is that we have lots of data. We're providing today data files that you can work with. So if you're not doing any of the experimental design and sample preparation and DNA collection and so forth, we can give you files if you just want to work on the analysis elements with your students, or possibly you can collaborate with colleagues of yours to do the analysis on their microbiomes. Okay, so just to kind of make sure that we're starting from the same place, we'll be looking at these FASTQ files. And if you're not familiar with FASTQ, essentially, if you look over here on the left, in your FASTQ file, there'll be thousands of reads, and they'll all have the basic format. There'll be a sequence ID, there'll be your actual sequence, and then there'll be a line here on the fourth line, which is a bunch of characters, which indicates the quality of the read at each position, okay? And they'll generally come if you're getting sequencing from a sequencing facility or off your own sequencer. You'll have, your samples will have their FASTQ files. This .gz just means that the file's compressed. And so these are generally the starting point for Nephele. If you're doing single end reads, you're going to have, again, a sequencing primer that's going to read into your insert in one direction. In this case, the insert is the amplicon from the 16S gene that we've amplified using 16S primers. If you're doing paired end, you'll have two reads coming from both directions, and then those reads will be merged into a single read. So with the paired end read, you get a little bit more data, about 250 base pairs relative to the single end reads. So that's kind of what we're starting with. And again, if you're interested, those crazy characters align with the Fred scores, which is the quality scores. But just as a shorthand, that if your Fred score is above 10, it's greater than 90% accuracy. If it's at 30 or above, it's 99.9% accuracy. This can be a bit important when you're choosing the platform, because the Illumina, your Fred scores are probably going to be close to 30 or above. But if you're using Oxford Nanopore or MinION, they'll probably only be in the 10 to 12, 14 range maybe. And so those differences in accuracy make some kind of impact kind of the questions that you can ask. All right, and like I said, oftentimes when you get the files, they're going to look like this. And if you're doing paired end reads like we'll work with today, you'll have two identically named files, and it'll just be R1 for read 1 and R2 for read 2. Other than that, they'll be identical. So if you have 12 samples, you'll have 24 FASTQ files. All right. So the first hurdle that you need to get over with the analysis is to create your metadata file. And for those of you that have maybe done next generation sequencing for several years, you might have heard of a mapping file and a metadata file. For what we're going to do with Nephele, you really only need the metadata file, because the mapping file usually tells the program where your data are, but you're actually going to point your browser to where the data are, so you don't need the mapping file. So in the terminology, sometimes they might refer to it as a mapping file, but really it's just a metadata file. And you can see an example of a metadata file here on the left. It's just an Excel spreadsheet. And Nephele requires that the format kind of follow this sample ID forward FASTQ file. This is for a single end file. I'll show you a paired end read in just a minute. And on Nephele, you can download a template that kind of shows you exactly what you need. And that template here is shown on the right. I'm just going to pause again to see if there are any questions. Everyone's doing all right? Okay. So here's a metadata file for a paired end read. And again, I've indicated with these arrows the columns that you're required to have if you want to submit your samples into Nephele. Sample ID, forward and reverse reads in columns B and C. There's a requirement for a treatment group. And again, depending on the project you're doing, you may not have a treatment group, but you still need to create a column called treatment group and put some information in there. And so I've just kind of given some information about my samples in the treatment group, even though in this particular case there wasn't a treatment. And then I've created all the other columns, and these other columns can have whatever information you think is relevant to your samples. So I have sample type, the date collected. These are soil samples, so I have depth, who was there collecting. I have lat and longitude. In terms of what to put in the metadata file, I encourage you to put more is actually kind of better. There might be information that you might not use at first, but then you decide, oh, that would be interesting to look at that. If you don't have the metadata when you first submit, then you need to kind of go back through the process with that new column of information included. So if you have it all there to begin with, that can save some time. So the only thing I would hesitate to put in the metadata file, if there's any privacy issues, if you have patient data or something like that, you would want to keep anything that's identifiable out of the metadata. But other than that, pretty much anything is good to go in there. And again, here you can see those file names. These are the file names from my FASTQ files. And you can see that all the forward reads, all the R1 reads are in the forward FASTQ column, and all the R2 reverse reads are in the reverse FASTQ file. And so that's the way it has to be formatted, and the syntax is important. The columns have to be exactly like this. I'll show you in a little bit. When you submit your metadata file, Nephele does a kind of a check on it, and it'll let you know if there are any errors. And I can talk about that in a little bit when we do that. Right, so I'm going to try to run things live, and you can follow along on Nephele if you want. If you're accessing, you should be able to access the same files from our folder that I'm going to be using. Hopefully it's not going to overwhelm the system. But again, you don't need to follow along because these videos will be saved and you can do it on your own time as well. And okay, so let me exit out of here. Oh, actually, I probably didn't need to do that. Right. Can everyone still see my screen? Okay. So this is the Nephele homepage. It's hosted by the NIAID. And if you just Google like Nephele microbiomes or Nephele NIAID, it should be the top hit. I encourage people, if you haven't already had a chance, to create an account. So you can see I'm logged in. The accounts are free. You can have your students create accounts, but it gives you a little more kind of ability to use some of the functions if you actually have an account. And it may at this point force you to create an account. I can't remember exactly what the setup is now. They've changed things a little bit. In general, the Nephele divides the microbiome analyses into three main steps. Step one is pre-processing. That's what I'll show you in a couple minutes. Step two is analyze. And then step three is explore. And we'll talk about steps 2 and 3 probably in the next session. Just a couple important things to look at when you're here. We'll walk you through what we do, but Nephele has lots of good support. If you look under the user guides or the tutorials, there are lots of good support information on how to use Nephele. If I click on my workspace, I can see all of the jobs that I've run over the last 90 days. So this is a kind of a good way to keep things organized, at least for a little while. Once that 90 days has expired, you need to have downloaded the data outputs into your own folder. Okay, so let me start by going through the pre-processing steps. So I'm going to click here on pre-processing. The other thing that I'll point out here is that depending on where your sequence data are coming from, they may already have gone through pre-processing. So if you use like Mr. DNA or Wright Labs, the FASTQ files that you get have already been processed. They've been quality checked, they've had the primers trimmed, and they're ready to go. So you don't necessarily need to do the pre-processing. And the data files that we're working with today are from Wright Labs. So they have already been processed. I'm going to show you what you would do, but we're just going to stick to all the default settings because there isn't actually anything to process in the files that we have. So what you'll do here is you'll click on Start Job, and that'll get you into the pathway to work with your samples. Like I said, we're going to do paired end, so I click on paired end, and then I'm going to click on Next here. And my files are in the Google Drive. You can have them local, or you can use some of these other options, but I generally use the Google Drive if I can. And see, it's okay, a little bit slow, but hopefully we can manage. And Davida always yells at me about my all my stuff in here. So here we are in the folder, and I'm going to go to the FASTQ data files. And so here are all those FASTQ data files that we're going to work with. And so I'm just going to hit continue because those are my FASTQ files. The next thing that it's going to do is ask me for it's calling it a mapping file. I told you it's not really a mapping file, it's a metadata file, but anyways. So I'm going to go and find my mapping file, and I think, where is it? Here it is. Okay, so I'm going to click on that and upload. Okay, so once you've done that, it gives you a few options here. Some you don't have any choice, so it's always going to do the FastQC, and it's always going to give you a summary. But like I mentioned, if your data have not already been processed, you would want to trim primers or adapters. If you haven't already had your data go through a quality a screening process, you would want to do that here as well. Like I said, these data have already been through that, so we'll just leave those unselected. And I'm going to click on select here. And so here is where I can put in a little bit of information on the particular job. So I did this morning when I was just checking to make sure everything was running. So I'll call this QC2. I'm not going to check on any of these because we've, like I said, already done that. And what I'm going to do is to click on validate and submit. All right. And so this is the way I'm showing you this for QC, but this is the way it works for all of the different steps. It's now telling me that this job has been created, and it's sending that information to the email addresses associated with this account, which is my email address. So in my inbox, I'm going to get an email in a few seconds that says that this job has been submitted. When the job is complete, I can go to my workspace. And so here you can see the job that I just submitted, and it's in the process. And when it's complete, it'll tell me here. But I will also get an email when that job is complete, and then I can go and look at the files. A couple notes here. If you're doing this in a class, we usually do the cooking show format like we're using here, that I've already run these, because depending on the size of the data and how much use Nephele is going to use, how much use Nephele is getting, this can take a few minutes, or it could take a couple hours. Usually it's a few minutes, but if you're doing this in a class, obviously you may not want to sit around and wait for 20 minutes for the files to come back. So depending on how you're structuring things, you may want to have done things beforehand. The other thing that I wanted to point out, let's think of where the best way to show it is. Let's see here. So actually, maybe that's not the. This is the metadata file again. Okay, so this is the folder where all the FASTQ files are. And in that metadata file, every single file that you're going to analyze needs to be in the folder that you're pointing Nephele to. If there's if you say in your metadata file, look for sample 17, and sample 17 is not there, you're going to get an error. However, you can have a lot of other files in here as well, and that won't mess things up. So initially, when I was doing this, I kept making sure that what was in this folder matched exactly what was in my metadata file. But then I figured out that you can have extra files in here that might not be related to the analysis that you're doing, and that's fine. So you don't need to worry about matching it up perfectly. You just need to make sure that every file that you list in the metadata file is actually in the folder that you're pointing to. Okay. The other thing that actually go to that I wanted to point out is when you're at the pre-processing. So I told you that we weren't doing a bunch of steps because they've already been done. If you want to know more about those, again, I clicked on the more information, I think. What did I click on? Yeah, learn more. This tells you how to do the adapter trimming if you need to do that, how to do the quality trimming and the merging if that hasn't already been done. And so again, like I said, there are lots of good resources here that'll walk you through how to trim the primers, how to do the quality trimming, and so forth. Let me just pause again. Any questions out there? Or
there was a good question about adapter trimming, that if you were getting your sequences and you didn't know if they had been trimmed or not, would it be a problem?
No. So I've done that several times. I'm just not doing it to save a little bit of time today. But it won't cause any problems. You can do all of these steps on the data, and it's fine. And actually, sometimes when I've done this, it will actually, on the quality trimming, it will throw out a couple that were not thrown out by the quality trimming that was done by the sequencing company. Usually, it's just a handful, and that's fine. The one thing that I'll point out that's important is that so we pointed to the raw data or the FASTQ files that I got from the sequencing company when we went in to do the QC step. The QC step output will provide a file if you've actually done any of this trimming and quality analysis and so forth. We'll give you a new folder with all of those FASTQ files with the trimming and so forth. You have to remember that when you go on then to the analysis and the explore step, that you now point to that new folder with your trimmed FASTQ files and not back to the original one, or else you're running it on, you've just kind of short-circuited all the work you just did. So and I've done that, so it's annoying. So just remember that if you are actually doing the QC step, to look at those. Crystal, you have a question?
I understand all the trimming and such, but I was curious about this paired end read merging. Would it leave us, instead of having the two files, with one now that it's been merged? And so that's what we'd be using?
Yeah.
Mm-hmm.
So I think there'll be a folder that says like merged.
Okay. And so if we chose Nephele to do that for us by clicking those boxes that you didn't this time, would I still supply it my two sequence files?
Yeah.
And then it would create the one and do
Yes.
that all behind the scenes, and then we'd go on to analyze that merged read.
Exactly.
Thank
And
you.
if you look at, I mean, you can click on some of these things and it tells you more about exactly how it does the merging, and that it, like, I think the default is that it has to have a 35 base pair overlap between the forward and the reverse reads, but you can change a lot of these things. But generally, when we just get started, we stick with the defaults. Other questions out there? Okay. So what I'm going to do now is, let's see. So I'm in the webinar folder here, and I have a folder here that's called Nephele outputs, and I'm going to click on that. So I don't know if the output that I just ran came back yet, but this is one of the ones that I've done previously. And on my computer, when these come and I download them, it just goes to my download folder called, output 10, output 11, output 12. And I usually just rename those so I know what they are. And then I upload them to my whatever folder I'm doing the analysis in. And so in this case, I have outputs, and it's from the QC demo. And so this is the file that, Crystal, you were just talking about, that if you do those things, all of those QC'd FASTQ files will be in this output folder. And the other thing that's important in this folder, so let me just step back a sec. So for all of the steps that you're going to do, it's going to generate a log file. And this is really important. If things go smoothly, it's a little bit less important. Here you can see the log file here, and it's essentially telling you everything that the QC program did. And of course, a lot of it is just the same thing over and over again, and it probably uses terminology and stuff that I might not fully understand, but it's telling me that everything went okay. If something didn't go right, if there was an error and you open this log file .txt, it'll tell you where the error was and at what step. And oftentimes you can kind of understand it yourself and figure out what the mistake might have been. But certainly, if you've hit this as a roadblock that the QC or the analysis or the Explorer didn't work and you can't immediately figure it out, you can contact us or you can contact Nephele, and they'll want this log file because that's the way that they'll be able to figure out what went wrong. So just I'm pointing it out that it's an important file to kind of keep track of how things went. And if you do get stuck and you go, to someone who knows a little more about QIIME 2 and Nephele and so forth, they'll want this log file. But what I'm going to show you now is the QC report that comes out. All right, and hopefully everyone can still see this. So this is the standard report that I'm going to get on all of the FASTQ files that I submitted. Okay, and this general statistic, well, so first of all, I'll point out that there's a helpful little tutorial video you can watch if you want to learn more about the QC steps. Right here in this dashboard, it's showing me the number of duplicates in each sample that I submitted, the GC content, and then most importantly, at least for us, is the number of sequences. So in this right-hand column, I can see how many sequences got read in each of my samples. So my negative control doesn't have very many. That more or less makes sense because there wasn't anything really there to sequence. Some of them I have 25,000, 50,000. A lot of them worked really well. Some of them, there must have been, you know, bad sample or something went wrong, and I only have a few thousand. But you can see how many sequence reads you get per sample here, and that's oftentimes important to know if you want to figure out, did my run go well? Did most of my samples, you know, hit my expected threshold of 10,000 or 20,000 reads, whatever it is. So there you can, that's helpful to look at. The other thing is there's a lot of data that I don't necessarily need to look at in the QC report. This is showing the number of duplicates. Again, for us, we expect a lot of duplicates. So if obviously every time it reads a sequence from the same bacteria, and if you have a bacteria that's present in your sample and it makes up 50% of the sample, obviously you're going to have a lot of duplicate reads from that particular bacterium. So again, you shouldn't be worried if you have lots of duplicates. It's not a bad indicator, even though down here below, I think it'll tell me that my reads failed because there were too many duplicates. The other thing I'll show you is that if you want to learn more about this, you can click on help, and that gives you a little brief description of what each of these is doing. What I like to look at after that initial dashboard is the histogram here that shows me how my samples looked. And here is getting back to the Phred scores. Remember, if it's a Phred score of above 30, that means it's 99.9% accurate, okay? And you can see that most of my reads, this is the average Phred score at each base pair, and most of my reads are about 250 base pairs. And so I can see that most of them stay well above that 30 Phred score. There are a few that dip down, but most of them look really good. So that's an indicator that it was good quality DNA and things went smoothly. The other reports that it spits out, again, are not necessarily relevant to microbiome sequencing. So this per base content, you know, it's telling me that 52 samples failed because it has some expectation that we're not actually aiming for. Davida, what's up?
So we have a couple of questions that I think will get lost if we don't stop now.
Okay. Yeah, sure.
One of the first question was, how do you select a threshold of a number of good quality reads with good QC? So I believe the Phred score will tell you that, but do you want to expand a little?
Okay, so yeah, so if you go to the in the QC pipeline, you can set the threshold for the Phred score there. I think I was just, you know, saying that my expectation, like when I send our samples to one of the sequencing companies and they say, well, how deep do you want us to sequence? And I'd say, well, I'd like to get 20,000 sequences per sample. And so I think when I was saying threshold in that context, I just meant that most of my samples, I expect that I should have 20,000 or more decent quality reads. But when you're doing the QC filtering step, again, if you're doing Illumina, you probably want to set that threshold. I think the default is like 27, but you could crank that up a little bit. If you're doing Oxford Nanopore, the Phred scores are much lower. So you'd have to reset the expectation to 10 or 11 or something like that. And we'll talk about that when we get into the Nanopore data in the fourth webinar. So the threshold I was just mentioning a second ago just meant my expectations on how deep I told the sequencing company to sequence. Does that answer the question, or did I not understand it?
I think that's it. And then we have a second question, and I think it related to when you were showing the Phred score chart. What is the 10 in orange, and should they be removed?
I
didn't quite capture what.
Right here?
Yes.
Okay. Yeah, so thankfully nothing's down as low as 10. So the lowest one looks like it's in the mid 20s. So again, this
sorry, I was asking, there is a bar next to sequence quality histograms, that one.
right
Yes.
here. Is that what you're talking about?
Yeah, because that refers to those curves in orange that are upper and lower with a lot of up and downs.
Right. So it's giving me a warning, which means that those samples had a lot of slightly lower quality reads, but it's not so bad that I wouldn't go forward. Remember that these are still going to be, I think if I'm remembering correctly, over 99% accurate. But again, I don't want to jump too far ahead to the nanopore stuff, but again, if you're only looking to identify your sample at the level of phylum or class or order, you might be able to get away with Phred scores of 10 to 15. If you want to be confident at the level of genus, then you really need to get your Phred scores up above 25 or 30, okay? So it's just all about how confident you are in what is going to be determined in the next step when QIIME 2 tells you that these are the taxa that it finds in your sample. Okay, so again, some of this, again, is going to depend on the platform that you're using. Does that answer the question?
Yes,
I
thank you.
thumbs up.
And,
Okay.
and I just put another question. Nanopore doesn't give you that many reads, because of the system they use. I was expecting the curve not to be this smooth and with a lot of up and downs.
Yes, and again, yes, with Nanopore, you're going to get fewer reads and they're going to be lower. Well, that's not necessarily true. I mean, if you're using, again, depending on how many barcodes you use and so on and so forth, but generally speaking, yes, it'll be fewer. The upside, which I'll kind of a teaser for week 4, is that because the reads are so much longer with the Nanopore, you get close to 14 to 1600 base pairs. The fact that there are more errors, you still have a much longer read, and so that allows you to still do a lot of really great stuff with it. So again, here we have 99.9% accuracy, but we only have 250 base pairs. With the Nanopore, you're going to have closer to 1500 and lower quality, but it can kind of even out a bit. All right, so we're kind of getting, I got a little too talky here. I'm sorry about that. But before, I don't want to abruptly end. So let me stop here and make sure that I have a chance to answer any questions that I might have missed. Anything out there? So one thing that I want to ask, and I don't know if Rachel set this up as a poll or not, is that in preparation for week 4, so we know from when people apply that there's a handful of you, maybe more than a handful out there. Oh, here it is right now. Okay. Who would like to consider using or already using Nanopore. And so we want to kind of know how to tailor the session 4. So if you can respond to these questions here in the poll, that'll give us a sense of maybe where we should put emphasis for session 4. And session 2, I apologize if nothing seemed really fun here, because this is kind of like the nitty gritty, just learning how to get things into Nephele and so forth. But sessions 2 and 3 are really cool because we actually get to look at the data and see some of the visualizations. And so please do make sure you come back for those, because those are going to be hopefully more exciting. I mean, I find all of this exciting, but I can't speak for everyone. Any questions out there before we wrap up? I think you have our contact information. If questions do pop up within the next 14 days or so, go ahead and send me an email or send David an email, and we'll do our best to answer your questions. How are we doing, Rachel?
Second poll in there. So different questions, looking at whether you think you're going to go to Oxford Nanopore in the next year. So 2 more questions. So that makes 5 quick yes, no questions. And then can you put your email into the chat for folks? So that if they have questions about contact, they can contact you. That's great.
I'm sorry, is Oxford Nanopore as opposed to, say, Illumina or PacBio? Are these the sequencing platforms that we're?
Yeah. So
okay.
we're I only have experience with Illumina and MinION, the nanopore system. I know about the technologies for PacBio and Ion Torrent, but I've never worked with those data, so we're not going to cover those, other than just mentioning that they exist in these webinars. If there's enough interest out there, then we can try to up our game and think about doing a webinar in the future that would focus on something like PacBio, but right now we just don't have the expertise. And if you recommend it, then you might be our expert.
No, so we would do Illumina. So I'm wondering the PacBio question, I'm sorry, the Oxford Nanopore specific questions. That's just a different technology?
It's a different platform, it's different technology. I would recommend, even if you're doing Illumina, because 99% of what we've done to this point is Illumina, but we're also now doing Nanopore. There are some pros and cons of both, but the Oxford Nanopore is so accessible and it's so field deployable that my sense in looking forward is that a lot of people that are working with students are going to migrate to Nanopore. I could be wrong, but that's kind of the trend I see. All right.
So we're at top of the hour and time's up. So thanks for attending, everybody. So recording of this will be posted within a day onto our course LMS. Our next event will be in 2 weeks, same time, 2 p.m. Eastern time start, and you'll access that webinar through the LMS as well. So remember, if you have questions about content, you contact Theo, and if you have questions about logistics, you contact me, and there's my email address into the chat. Okay, thanks for joining today.
Thanks, everyone.